Ever since Darwin, biologists have understood the importance of the tree of life metaphor. In Phylometh, we will learn how to infer that tree and how to use it to understand biological processes. Phylometh is made possible through a career grant from NSF, as well as ongoing support from the University of Tennessee, Knoxville.
So we're talking about APIs, a way for computers to talk to other computers, right? And so here's open trees. But let's talk about the general interface. So like the New York Times, right? So here's a New York Times article. Here's a URL for it. And here's the URL bigger so you can see it. So what do you see about it? What do you notice?
Has the title of the article in it.
Yeah, it has the title of the article in it.
That's how it works.
Yep. Plus arguments, right? And this is stuff like, where did this person come to the article from? They got it from the homepage. Right? Where they click on? They clicked on the headline, right? Things like that. And also this stuff, right?
That's the actual document.
As a document, right? So I could just use just this to get to the article without all this other stuff, right? But also you can sort of see, if you want to look at New York Times articles from 2016, how might you do that? Right, something like this. Right? Why did you think that?
Because of its structure.
Right, because of the structure, right? And so a lot of websites are structured like this. Now, will it actually work for the New York Times? I don't know. Oh, let's try. No, it's not work for the New York Times. But for many sites, it will.
And
Right.
maybe internally.
Probably not, actually. And so once you have that, you can start figuring out, you can start navigating websites that way. And so that sort of interface is called a RESTful interface. So just like how you interact with a regular website. You can enter in information, figure out how to go back to the home page by dropping stuff after the slash. And so the Open Tree of Life API is very similar to that, right? Where you have, opentreeoflife.org, and if you do opentreeoflife.org/treeoflife/about, it gives you information about the draft tree. If you do /mrca, it gives you most recent common ancestor. If you do subtree, etc. Okay, and so the nice thing about this sort of interface is it works similar to the way the regular web works, right? So you can see how it looks like the New York Times URL, right? And so you can get information from this. There are other ways of doing APIs that involve, hidden parameters you pass and things like this, but a lot of them are now moving towards this sort of format. Okay. And so, for example, it makes it easy if you want to scrape something. So if I knew, let's go to an OpenTree page. Okay, let's search for taxon. And it's on this study page. All right, so here's Pyron et al. 2013 as a study. Let's look at a different study. How could I figure that out? Well, I could try doing this from 2460 to 2461. What's there? That's a different study, right? So already you can see if you want to take every study off their website, you can write a little script that just goes from PG1 to PG large number and download each page, right? Because they have a very simple structure to this website. Okay. If you were to do that in real life, you should first check with this website, because if you start doing that a lot, it makes a lot of connections. You actually bring a server down. That's happened in the past.
So you can use a function though that scrapes just what you see from. You don't have to download the metadata for the whole page.
It depends. I mean, so there are lots of functions. So rvest is a new package in R for this, and you can say, given the structure of the page, I want to go just into this field here and get just that field. Or you might have to download the whole page and write something that uses a regular expression. So a way to say, match these characters. So match, taxon equals some sort of text word, right? And so I can search for taxon equals word, and then it will give me, anolas, formica, myrmecystis, right? So that's a regular expression. Okay, so you can search for these patterns. Right, so it's like doing find change, but the thing you find can be, find me 5 digits and 3 letters rather than find me this exact string. Okay, so you can process pages that way too. Right. The main point for what I want to learn from this is just, given this structure, you could imagine making something that would, download all of this, or make something that would, search a list of studies for a name. Let's look for all studies that have Baker, and then go to that Baker page, right? And that's basically what ROTL does. It's what the treebase package does. Okay.
But if you knew HTML code pretty well, you could use that to your advantage when you're scraping.
Yeah. So let's look at this one. So
I'm looking at it. I wouldn't know where to start with. So.
Yeah. So in most web browsers, you can do just in the shortcuts. Show page source, either in Chrome or in Firefox or Safari. And here's what the page source is, right? And it's just HTML code, right? And the beginning is all this formatting information, basically. But as you dig down, you can get to oftentimes the content. Depends on how it's set up. So here, it doesn't have that because it's I think, keeping it as an image. But other pages you can. So if we do, that's a good one. I could do treebase. It's not a repository of trees. Search for taxon. Studies. Treebase is a little slow, as you would have found from doing your homework. While it's doing it. So here you can see the actual structure. So you see this. It's a table in HTML. Let me make this bigger. Right, and you can see, repeated things, right? Input type, text box, something here, and then taxon name, right? Again, input type, text box, something here, taxon name, right? So you can go through and get this feel of what the structure would be, okay? And so all these interfaces that we're using today basically do this sort of thing. Okay, so for OpenTree, they have these sort of interfaces to it, right? And you can get information back. So I can do this call, so I can go to basically just this URL, right? And it'll give me back this information from OpenTree of Life. Okay, I can do MRCA for possibly a set of node IDs I know. So go to node 17 on your tree, and it'll give me back the MRCA for those nodes. Okay, and that's all that this R package is doing. All right. So any questions about that?
Where are you at right now? Where's that information?
So if I just search for OpenTree of Life API, and that's stored in their GitHub repo. Right, so everything they do is public, and so you can look at the documentation there. So as they update their information, you can check the latest version. Okay. How data come back to you? They could come back in a CSV file, right? Comma separated values, like you get from Excel. They can be in other formats too, like XML, right, which is related to HTML with behind web pages, but it's more nested. JSON, which is another way that websites give information back. It's not something you should need to worry about at this point, right? But so if you dig into it more, you can learn this. Okay. So for us, for homework, we had using ROTL and treebase, right? And so another question is with the treebase one. The ROTL, is that working for people now?
So all we had to do is just go to the GitHub webpage and
Right. So
that was just to get you in the door. So
yeah. So like everything else in the world nowadays, this has a GitHub repository, right? And so, and a README, okay? And so there's two things we could do. We could do install packages, and that gets things from CRAN, right? So we're used to that. When you install new R packages, you do that, go to CRAN. What's nice about that?
it's curated.
It's curated, yep. Why do we care?
So we know it all works as it is in that version.
Yes, we know it all works. And we know it all works in some ways, right? We know it doesn't so your credit card information, presumably, we know it doesn't throw an error. We don't know that gives you the right answer, right? So I could throw something on CRAN that says, has phylogenetic mean, and it just takes the first taxon name, and it gives you that, right? It would give you a number, but the CRAN people would not be able to detect that that's actually horrible and it's giving you the wrong answer, right? So they check for things, but it is a lot better than just random code, right? The problem with that, what are problems with CRAN? Demanding in what way?
To update things a lot, format it a certain way.
Yep. So they're very persnickety at CRAN, right? Which is great for users, but for developers, so we have code where we're using Diversity, which is a popular package, to test our own code to show, yep, it's still working, right? Same way you're doing a unit test in this class, we do unit tests in our code. Well, Diversity was going to get thrown off of CRAN for a little while. It's like, oh, you can't do unit tests using Diversity. Well, we want to check it. So they're like, you either throw out the tests or do something else, right? It seems like that sort of problem. Whereas GitHub, anyone, you've all posted things to GitHub now, right? Would you want someone to rely on your code to, do a study of cancer evolution? Hopefully not, right? But you could let people download it and use it, okay? And so GitHub is not checked in any way. Right? Just the code's up there. But for stuff that's actually developed, it can be a lot faster because creating to submit it, they have to look at it, they get back to you, you make changes. It's almost like peer review, right? And it doesn't take that long, but it can take a little while. And they also don't let you submit many times. If you're developing it actively, if you submit it more than twice a month, they'll get angry, right? So for this code, we can see it's being actively developed, right? So the R was changed 2 days ago, right? So you can go in and see what Francois is working on, right? So some utilities. Look at the history of this, right? And so we can see what he's been working on, the namespace issue, harmonized output, and so his little commit messages, you can see it. And let's just see, when he works on it, when other people work on it, and so forth, right? And so it's being actively worked on. So there was a bug in it, and I actually didn't. So when I first used this code last week to put it in, it was working. Sometime in the past time, I think OpenTree changed their API a little bit, and then now they fixed this. So Francois fixed this last night without me telling him. He just actively working and fixed it, right? So you can install the latest version yourself, but that hasn't had time to go on CRAN yet. So what you can do instead is install it directly from GitHub. And there's two ways to do that. You can download the package, like checking it out, or doing a git clone, or just downloading the zip, and then install from R directly. Or you can use this DevTools package, and that allows you to install directly from GitHub. Okay, so try that. Let's see what happens. If you don't have DevTools installed, of course you have installed DevTools. I think mine will fail. We'll see.
I did it without those arguments, and it worked fine.
Yep. So what do the arguments mean?
Oh, install dependencies. I should have already had those though.
Yep. And so dependencies are other packages it relies upon, right? So if this requires ape for dealing with trees, right, which most packages do, then if you don't have ape on your system, it will install it for you. Okay. What about vignette? What's a vignette?
Tutorial.
Right. It's basically like the R speak for user manual type things, right? But oftentimes they're focused on particular things. So if you want to use this package for doing this, here's a vignette for that. If you want to use it for doing this, here's a vignette for that. Okay. More and more packages are getting vignettes, but they're still fairly rare. Okay. So in my case, the vignette failed. Okay. And it has to do with how it's being compiled into a PDF. Okay, it's not a big deal. So I'll redo this command. I'll do build vignette equals false. And this is annoying because I don't have the vignette to look at on my computer, but I can still get it online. Okay, so now RTL is installed. All right. So any questions about that?
It says that it's done like it start RTL. Would there be any reason for it not to like recognize the functions like DNRS and names?
Yes, because it's been installed but hasn't been loaded. So you have to then do library ROTL. Because now on your computer, it's like Tyson earlier today, for me when I set up a new computer, I install all of Crayon on it. So I always have all the packages I might need, if I'm in the field or something. I don't go to the field anymore, but if I were in the field, I need to check the stock market, I could do it with R. But, I don't know you don't need to do that, but then, but still, not everything's loaded into memory. Right? So you have to tell it, use the library.
So somewhere in the package, which is our homework assignment, it tells it what the package is to load.
Yes. So
But it's not in just like the exercise.
Nope.
Document.
Because that's a good question. So the homework is actually a package, right? And so if we look at description, the text editor, you can see it depends on these packages: ape, treebase, ROTL, and suggests test that and filebase. Okay, which means that it'll automatically load those packages in, and more detail, it'll import functions from those packages. Right? So it's not actually doing a library load typically. If I just want to use one function from ape, just plot philo, I could just have it just load that plot file and load nothing else from ape. Okay. Instead of doing the simple way of just importing every function in all those packages. Right? What's the advantages and disadvantages of doing that? And basically what it's saying is doing a library ape, library treebase, library ROTL. So what could be the advantages and disadvantages of doing that?
If the function you're trying to download is dependent on other functions within the library, which are only downloading that one function, you won't necessarily be downloading the other functions.
It's not so much downloading; they're on my computer, but yeah.
Yeah.
Almost actually are smart enough to know that if it's an internal function, so if plot philo requires reorder philo, which it does, it knows enough to look inside its own package. So I would worry about that too, but actually that's okay. Yeah.
Masking functions.
Yeah, what do you mean?
So if you have several packages that have a similar function, or one that have the same name, then one of the functions might override the other and be used instead of the one-to-one.
Yep, exactly. So they're basic functions. If the function has the same name, which one do you want to use, right? And so you can specify that by saying like ape ... reorder or filebase ... reorder, something like that, if they have the same function name. But otherwise, it's just whichever one you loaded last. Right? It could be a horrible issue for like reproducibility and things like that. Right? So you have your little script you're running, and you sometimes do library Abe, sometimes you do library Geiger first, whatever. If they have the same function name, you could have that happening. So sometimes it works, sometimes it doesn't work. Yep. Good. So that's the basic reasons. Okay. So yeah, for your homework then, it's automatically loaded. And so when you do R-command check, it loads what you need and runs it. If you're editing it line by line, though, then you need to do the library calls. Okay. If you're running it line by line. Does that make sense? Okay.
We're still getting errors for the treebase though.
Yeah, so we'll get to the treebase. Yeah, we're just the ROTL. Yep.
I've gotten an error when I try to do library ROTL.
Okay.
It won't.
It says package name space load fail for. Yeah. No. Yeah, that is like.
All right. So what you can do in that case is try quitting R, open R again, reinstall, and then library.
It's just here. So that's good. That's what good.
Okay. So make sense now? And so you've done the exercise for ROTL. You can see how it works. The thing to remember about Open Tree of Life is it has two sets of trees. Well, three sets of trees, actually. One is its giant super tree of all life, right? And they just got this published in, I think, PNAS, and it's huge, and it's very, very unresolved, right? And the way they create, I mean, look at this tree of Anolis, for example, right? I mean, they did not have one speciation event that led to, 200 Anolis species, right? It's just lack of information on the tree, right? And part of it with the way they create the tree, they use a mixture of, peer-reviewed trees that are published and taxonomy. Right? Why would you use taxonomy for this?
Maybe like as a guide to be quicker to make the tree, so that you automatically say, we start with all anolis where we think anolis should be, and it won't try to put them where elephants are.
Right.
Waste time with them.
So partly, if you're doing like a tree search or something, you can have a guide tree based on taxonomy that will keep your tree from trying to put things in weird places, right? They don't actually do a tree search per se, but that's something you could do. And one danger with that, of course, is that taxonomy can sometimes be wrong, right? And so then if you constrain it to match the taxonomy, you won't be able to detect that it's wrong because you just gives you the same answer, right? In their case, they're not actually doing constrained search so much. We're doing more of a consensus type thing. So why else might you want to use taxonomy?
To make the Claytus happy.
You can't. It's impossible to make Claytus happy.
Oh,
Yeah. Well, I mean, but what do you mean?
I don't know.
Yeah. I mean, it helps you get a better answer, right? I think if you constrain the search, you could, but they're not searching. The goal is to get a tree of all species, right? Are all species in GenBank? No. What fraction are? 10%? Will agree, disagree?
5%.
5? 10% is right. So, yeah. Out of the known described species, right? So of course, there's always canopy fog, a new tree in the tropics, you get lots of new species and stuff. So we don't know exactly how many species there are. We look at the named species, and the named species in GenBank, it's about 10%. Okay, so the rest of the 90% still matter for life, right? If you want to understand diversification or history of life, you need to know where those species go, right? But there's no genetic data to do it. There's no morphological data for those either, right? But what there is though is their name. Right, so I have this identified species. I know it's in this genus, which is in this family, which is in this order. Right, and so the taxonomy gives you that level of nestings. So they're assuming that Anolis is a clade. So all these things we don't have sequences for must be in that clade somewhere. And so that's how they construct the tree. That's why they have taxonomy. So OpenTree has this synthetic tree of everything, plus the source trees that went into it, plus the taxonomy. Okay, and so ROTL can give you any of those things. Now, for most of your use cases, in science, I'd argue you should go for the source trees, the real published trees based on data, right? The synthetic tree will be very, very unresolved. It won't have any branch lengths. And it has other problems with how they do the nestings and things like that, right? So you have to do a tree on a subset of taxa that are in existing trees. Now, one annoying thing is that OpenTree and treebase are not a one-to-one match. So there's trees that are in treebase but not OpenTree. There's trees that are in OpenTree but not treebase. So you can look in both places. And the trees that are in dry that are not on either of those two. And 96% of trees are nowhere. So we did a study a few years ago to see out of published trees, where do they go? Mostly nowhere. Mostly it's a JPEG on a paper. Right? There are R packages and JavaScript, Java apps and things like that allow you to steal papers from trees. There's one called Tree Thief. There's one called Tree Snatcher, where if you see a paper, you can trace the lines, right, and get a tree that way. Mark McPeak did a cool study years ago. We took a ruler and to measure branch lengths. He did 3 centimeters, 1.3 centimeters, and stored all those trees. So he's a database of that. Right, but general trees aren't available, right, which is annoying, but that's the way to play it. Okay. So questions about that? Yeah. All right. So let's talk about what happens when things don't work. Right. This might happen to you again in the future. I will. Right. So the tree-based thing. So this R treebase is part of R OpenSci. So what's R OpenSci? Yeah. So R OpenSci is a consortium of students. It was started of scientists. It started when they were postdocs mostly, but they're still all fairly junior, so we're now faculty and stuff. And basically what they do is they create these tremendously useful packages to get data from places. And it's got a big bunch of money, which made a lot of sense, actually. And so, yeah, just go look at the packages. Okay, so if you want to get data from Figshare, right, you can get that. If you want to get data from AntWeb, right? If you want to get gender, so I've used this to look at bias in salaries and bias in publication rates by people. So if you don't know people's gender, you can take their first names and estimate when they were born. It'll tell you, oh, that Pat is probably male, that Pat's probably female. Brian's probably a male name, Brenda's probably a female name, because things change over time. So you can just do that assignment, right? You can pull data from Neotoma, so paleoecology data, or PaleoDB, Dryad, right? So there's FishBase, GBIF, okay? So they have tons of these packages to get stuff out of from elsewhere, right? Fixing taxonomic names. Okay, so treebase is one of them, right? So go to the GitHub repo for that. Okay, and here's the source code, right? And so I tried it a while ago. It's going to be working. It's working right now. Okay, so what we can do is download stuff, try to figure out why it's not working. One warning sign. So see this thing here, build failing. What does that mean? Yeah. So we're doing this unit testing in class, right? So what does that mean? Why are we doing unit testing for our homework? So you run your check and it says, 2 out of 2 tests failed, 2 tests passed, or 4 tests passed, right? What are we doing in that case? Well, why are we doing that?
Seeing if the bits we wrote work.
Right, seeing if the bits they wrote work. And how do we know that they work?
They pass.
They pass what?
A check.
A check that I wrote, right? And eventually that you can write yourself, right? So for the alignment thing we're doing with math, right? We have data, They want to see if when you do the mapped alignment of data, do you get back data of the right length, or has it inserted too many gaps or deleted some stuff, right? And that's a very simple check. Okay. And what a lot of people do with GitHub is you can actually have someone else called Travis check your code for you, so they can run those tests for you. So if you enable that in your homework, every time you made a commit to check it, rather than having run checks yourself, this other group would run the checks for you and let you know, yep, passed, nope, failed. And that's what this little button's showing. It's showing that it's failed. Okay, so it's kind of handy to see what things going on with that. Okay, so let's install this. We have treebase, installing treebase. And what I should show you today is not only how to fix it, but how to, if something's not working right, try to figure out what's going on. Right? Is it you? Is it your connection? This is going to happen a lot in this class and in general. Right? So library treebase. Okay. And now what I can try doing is running some of, so here's their little help file. I can try running some of the things in their help file. Just see what's going well. So phylogeny is from Hulsenbeck. Right, so John Hulsenbeck's a great scientist who's at Berkeley. Posts a lot of trees and the cool stuff with long branch attraction and Bayesian phylogenetics and things like that. Right? And so now I'm trying to use their code. They're called. All right. So what do we see is happening here? Does the red mean we should worry? Sometimes, right? Not all the time. It's like loading required package ape. That's fine. We like ape, right? Error in scan can open connection. That's an issue, right? Okay. So see it failed, right? And we can just verify that by we're supposed to store stuff in this object. Let's see. Empty list. Okay. Now presumably their check used to work, right? Their little example code used to work. Now it doesn't work. Right? How do you figure out why it doesn't work?
Hold on. You said the phylogenies from Hulsenbeck was a example data set or something?
It's so here's the function we used. Phylogeny from Hulsenbeck is stored from search treebase, which comes directly from their help manual, right? But it failed, right? And how do you know it failed? Well, we know it failed because first of all we got lots of errors. Sometimes you get errors and keep running though. Right? But then it should have had presumably phylogenies from Hulsenbeck, which should be stored there, right? And nothing, nothing came in. Okay. Now it could be, Wi-Fi is down, so you can check that, right? It could be treebase is down, so you can check that. It could be something wrong with the package itself. Maybe treebase's API changed. Okay. So you're trying to debug, you can start to dig into things and look at what's causing it. So one thing I like to do is actually look at the function, right? So if I do, question mark search treebase, we get help, right? So you know how that works. If I do search treebase like that, right, it just runs the function with no arguments, so it'll give me errors, right? If I do search treebase like that, no parens, what will happen? Gives you the code. Right? That's cool. I mean, R is so open. It gives you that and shows you the source code. Right? So it's an easy way to dig into it and see what's happening. Right? And at first it will look, scary, intimidating to see like this code, especially if it's written badly. Right? This given who wrote this is probably written pretty well, actually. So you can see. So this already tells us a lot. Right? So input, what's the default for input? What? There's no default, right? So if you're writing a function like this with just input, then you have to pass some input into it. Versus things like exact match equals false, right? Meaning that if I don't tell it exact match, it just defaults to being false. Okay? But input has no default. By has no default. branch lengths has a default. Okay. So does that make sense? Yep. Okay. And so I get a bunch of terms, right? So in terms of parse treebase by, so that's yep. So we can search by author, taxon, subject, etc. Right? For each one of those terms, it does something. So it stores it as a list, right? A vector actually, and then depending on what you pass it in, it will look for different things. Okay, and you can scroll through, and it's doing what's gsub? Anyone know what gsub does? Yep, it's find change, right? So all this is saying is find this plus and change it to this thing, which I think is like a space or some weird HTML character, okay? And do this, and then do this, and do that, and so forth. So for example, I could do, you know, what else to do? So pattern, replacement, and then what we're going to do it over. Right, and change cat to bat. Right. If you pass it in vector, it would do over the whole vector. Okay. And then we see the query it's doing, right? So again, since we know how APIs work, we can see, oh, it's calling this, and then the search type, the search term, the input. It's like we were doing the New York Times 2016 or 2014, right? Just passing that as a string, and then gets Nexus files from that. So this get next function is doing the heavy lifting.
So I don't see the scan function in there, though.
Ah, good. So scan's causing a problem. We don't see it doing scan, right? Which means it's probably somewhere nested deeper in, right? So probably in this get next function. So we can dig in there. Okay. What I can do first is a very simple here's the function again. What I could do is try running again. So what I'll do is do this line by line. So here's the actual function. You do the function. And rather than have to pass our arguments, I'll just go inside and just run it as a regular, so like you're writing a script. Right, so I have these arguments to use. Now let's take every time I see a return, a comma, I'll put a return in.
You're missing an I on the input.
What?
You're missing an I on the input. The top
Oh,
line.
yep. Good. So just find change for commas. This way we'll have all the arguments set up. Okay. So input, and I can look at my example function. See how this is a different font size? That's a bug in R's GUI. It happens all the time. Doesn't mean you're doing anything wrong. Just it happens.
I always go and change the font size of everything up and then back down so people can't tell I'm just copying and pasting.
Yeah. Let me save this in case R crashes. So I can go through and run this. Okay, so get curl handle isn't working right. That's probably so that's probably a dependency that we haven't installed, that we haven't loaded, right? So again, the package knows what to call, but we haven't called that library. So we can go and look at the description.
So this is what we would call debugging.
This is debugging. This is pretty intense debugging, but it's really to see how we sort of figure out what's going on. So it imports our curl XML. So let me just
There's no way to set the default in order to how to always paste things into the same original font size.
I don't know. And usually, actually now when I edit stuff, I usually don't do it in R. The advantage of doing R's editor is that, you know, if I have a function name down the bottom, it gives me the description. But the formatting things are weird. So I often now use Atom. I'll show you that in a sec. Let's try it again.
Why is it when you install packages, you have to do the quotation marks, but when you're calling up the library, they don't require it then?
Ah, yes. So when I'm installing a package, I could have a vector of packages, right? And so I could say, you know, sort of, yeah, so I could say, you know, cool packages. And so when I do install packages, if I do cool packages, it knows to use everything in that vector, right? But library calls one package at a time. So, yeah. Okay, so now I've gotten down part way. Here's an error here. Returns. See where returns is earlier up. So I only get so an argument like this should only return one thing, so tree. Try it again. So we could comment this out, just giving you basically what matcharg does. If I wanted to say TRE rather than TREE, it's only one thing that matches to that, so it would allow you to have abbreviations. Let's get rid of that. Cool. So now I have my query, and I can see the query, right? And so now that's what treebase package is trying to send online to get a tree back. Okay. Now what I can do is see, does it actually work? And so if it doesn't work, then there could be an issue with treebase, not this package. Yes, it's our RSS feedback through Chrome. Okay, so here it gave me XML back. And it has a species tree and another tree, right? So good. So we know this is giving us back something, right? So it's not now. It could be giving us back in the wrong format for this package, because you know it's giving back something. It's not that like treebase has turned off that aspect. Okay. So I'll go back to R. Now let's try the get next query. So you can't find the function get next because treebase doesn't expose that. Right? We talked about name collision and things like that. So some packages you can say, every function in the package is shown to the user. A lot of packages now hide lots of functions, and so treebase is one of those. We can say, show me anyway, treebase : : Okay, let's try running that one thing. Now it's running that. And see here, okay, here's where it's going. So I found two resources matching the query, looking for the Nexus files. Yes, this is where the error was, right? So now we've dug in. So notice not any of the stuff that's formatting stuff higher up is now somewhere in this function.
What would have happened if you would have tried to run that without the : : ?
That was the errors I was getting earlier. Could not find function get next because the treebase package knows that internally is not letting me as the user see it. Okay. So the way that's controlled in the treebase package.
Because I know that the input I have for like dev tools, it was dev tools : : .
So 2 colons means just go into the package and use this public function, right? But without having to load the whole package, you can just go in and just get one function. : : 3 colons is go inside and get this hidden function, right? So this treebase package exports only these functions, right? So even though there's other functions inside it, the only ones you can use easily are these. Okay.
And logic behind that is just like those are the utilities that people are going to want to use, and showing them more is going to be confusing.
Right. So if you look at, for example, I tend to be the view that we should expose more, but
Is it just because they're internal functions that make the functions work?
right. And you have to document them and stuff. So look at Geiger, right? That has a ton of functions, including ones that are like deprecated. Right? I mean, like don't use this anymore. I mean, look at the help for that. It says like stop, use something else instead. They've changed the function names, and so they have to write help for each of these, with some exceptions. And you can have a user go in and use it in some way. And so, or they could have a name collision, tips, could look with tips and ape or something like that. And so by exposing fewer functions, makes it simpler for the developer and hopefully simpler for the user, right? But for advanced users, this could be useful stuff that you can't get at.
But these are all exposed.
These are all exposed in Geiger, yeah. It actually has other functions. It has some functions called foo, with different numbers of O's that are hidden.
So
Stuff like that.
What about anonymous functions? Do things on CRAN use anonymous functions within their functions?
What do you mean anonymous functions?
Like that don't have a name and are just coded in the function, but then you can't call them in R.
Yeah. So you can have. Let me show you an example here. Let's probably do it this way.
It just seems like it would make debugging harder, if you're trying to define those.
Right. Loading it. So Here's the hidden function .const.foo. And it wouldn't be easy to see. So you can have other functions defined inside functions. I don't see any cases here where you just can't get at them at all. Yeah, so that can happen. That can make debugging harder. You can have things, there's a debug mode with question mark options, where you can go through, and if you hit an error, it can dump you deep nested within something. So if you're inside a function that has another function, you can see that function. But that's too high level for this point, right? What we're trying to do here is just show us, how if you have run into issues, how you can sort of dig into them. You can do it. And this is actually a harder issue than I expected it to be. But.
I was to say, I hope that you're like next week. I'm not expected to go do this.
No. But I mean, you see what the basic procedure, right?
Right,
So you try to say the error's here. Oh no, the error's now in here, right? Right. So what I can do now is open up this other function, treebase get next. Right. And what it could be is it's taking too long. Right. So let me try pause 1 equals 60 plus 2 equals 60.
It might just be bouncing back because it's
Too slow at treebase, right?
So are those seconds, minutes,
So I don't know.
clicks?
Generally in R it's seconds, but yeah, I guess we'll find out.
We're gonna get kicked
Is
out.
that some metric or English standard?
Yeah, they're not. No one needs metric seconds. There actually are metric seconds, like 100 seconds per minute, but yeah, no one uses them.
It's my favorite symbol.
Spinning beach ball of death is the technical term.
Or split R.
Let's see, it started at 35 seconds after.
When you're running big chunks of code, is the only way to know that it didn't crash to put little checkup things in your code, like 100 of 1,000 done, 200 of 1,000 done, or whatever?
Yeah, that's probably the easiest way to do it. And I mean that matters because sometimes R does just sort of get hung, and so if you're a user, you don't know if something crashed or if you should let it keep going for a while. Old school, so McClade, which is before you guys, had an animated cursor, and so like, instead of the spinning beach ball of death, it would have like a bug and then a palm tree and something else. And if you're really savvy, you could know what stage it was on and what icon is being shown. You could also do like progress bars, things like that. But, yeah, one handy thing.
How do you cycle through those in that view?
Oh, that's
Like, because those are not open programs.
right. That's a program installed. Quicksilver. It's free.
It's not.
Yeah.
Right. But it just works with the command tab.
No, it's a control.
Because it looks
I
similar
don't
to
know.
the command tab.
You know it until you have to say it, right?
Yeah, everything's muscle memory at this point.
It looks like it's the same interface.
Can't do anymore. You've broken me. There we go. Control space. Yeah, can't even like think anymore. Yeah, control space to bring that up, and you can just type. If I have a document I want to do, treebase.r or something, you can do that. It's super handy.
What was it called?
Quicksilver.
Oh, I think I already have that. Oh, I have something else.
Yeah.
Let's see.
Yeah.
Yeah.
Yeah, so R is still suffering. Oh, so it's running. So we'll see. But anyway, one thing I want to show is for progress while you're running things is this a modulus function. So %%. So if you do 500%. So basically the remainder. All this is just telling you what the remainder is. And so if we do this, the remainder is 0. If I do this, the remainder is 1, right? So if you want to just print out something every 100 steps, you can do step %% 100 equals 0. So if step equals 0, the remainder of dividing by 100 equals 0, print out something. Otherwise, don't. So I'm having to see, okay, this is exactly by 100 or whatever. You can just use that function. Right, so if you see something like Mr. Bayes where it says, print freak or something, or print f, how often you want it to print out each generation, it's using something like that inside. But, yeah. Okay. So that's still running. So we'll see if that fixes it. Treebase has been really slow. So Treebase has an interesting history. It was started by a paper by Sanderson et al. '94. And since then, it's been sort of bounced around. It was housed at Yale for a while, housed at Davis for a while. One guy who's been involved in it for a very long time is Bill Peel, who's now at the university in Singapore, I believe, who sort of keeps maintaining it. Before all, just like Treebase was a computer on someone's desk, right? So like the scale of what is like large-scale things in this field can be pretty small. I mean, GenBank is not a single computer on someone's desk, right? But you could download all of GenBank on your laptop; it would fit. No, not yours. But something like Treebase is very small.
About 2 terabytes.
Yeah, that's decent. Yeah. All right, so I want to see if this fixes it. Let's talk about other ways to get information for your trees and for your data. So one thing you can do, who's heard of Phylota? No? Okay. So Phylota was a project by Sanderson et al. It's still active. And basically what it does is take all of GenBank and blast it all against each other. So what's blast? Matching. Yep. So if I, go out and sequence something, I want to say, so I'm sick, they sequence bacterium in me, what is it, right? BLAST is a similarity search. We compare, a reference sequence, let's say a query sequence to a database, right? So we can say, okay, is it a plant or is it bacterium? Matches bacterium, right? It's most similar to E. coli 0157, also similar to these other bacteria. So that's what BLAST does. And so what this does is an all against all BLAST, right? So it takes sequence 1, compares it to sequence 2, 3, 4, 5, 18 million. Sequence 2, 1, 3, 4, 5. And so it does all of those and clumps things, right? And it clumps them into genes, okay? Rather than relying on gene names. Right? Why would you want to rely on gene names in GenBank?
Why would you not want to use it?
Yeah. Right, a lot of errors. So I could have, CO1, cytochrome oxidase 1, cytochrome oxidase I, which might not resolve to the same name. But they'd all be very similar sequences. And so that was their approach. And so what they do is, let's take, say, turtles. They group these searches and then group things by sort of genes. And then they record the number of taxa in GenBank for these groups. So if I was doing a turtle phylogeny, I could download turtle phylogeny from GenBank or something like that. But instead, I can just go here to turtles and look. See, they have about 400 species in GenBank for turtles. 472 descendant terminals, so it could be subspecies or things like that. Right? How many genera? And then sequence clusters. Right? So just a group of sequences that sort of could be a gene. Right? And found like informative sequence clusters. Right? So they have at least 4 taxa in them. Right? So 3 taxa arguably do have some information with branch lengths, but for inferring a phylogeny, 4 helps. So they have basically 178 genes that have information for turtle relationships. Right? It's pretty cool. So you can go there and look at this gene. What is it? So they take the first taxon in that set and tell you what it was called. So it's RAG2. Okay. And then how many taxon IDs are in it? 143. So you can look at that. And has 296 sequences in it, 143 different taxons, and it has length from 400 nucleotides to 1,400 nucleotides, right? And what they actually do is in the background, they actually do all the alignments for you and also make the trees, okay? The trees are done sort of fast and dirty because you can look at it, The alignments are also sort of fast and dirty, and they're not. And also, it takes a while for them to make them. So every new GenBank release, they update this. So the alignments aren't done yet, but we could just get the raw sequence data this way in faster format. So now we see, Caretta, and so these are all supposed to be the same gene for these, 200 taxa, right? And you can download that, run it through MAFT, like using the script we had from last week, and do an alignment. That's pretty cool.
It just pulls one per species or something.
I'm not sure about that. Actually, I think it does do one per species. No, it doesn't.
Yeah.
How's
It
that
like strips
affect-
away all the other information from it, right?
Well, depends. So that's you have a choice of use taxon names, use defline, use GI number. You can use all. This includes, yeah. And so that can be really handy for getting large, because we all know for our groups, oh yeah, everyone uses CF1 for this, everyone uses 28S. There's another gene that's used a lot, enolase, that you haven't heard of, but it could be really common for those taxa on GenBank, and so it allows you to pull those down directly. Okay, so it's very.
What if it's like multi-gene one?
So sometimes it'll be like 18S and 5.8 and 28S and stuff like that, and so sometimes it ignores the names. Sometimes you might find one section that has all three of those, and then another gene that has only one of those, and so.
Could you exclude those?
Yeah, but you have to do it visually.
Okay.
Yeah. Or you could split the one that's three genes into three separate things and then align them to the other 18S ones. Yeah. So it's not perfect, but. And as far as I know, there's no R package for this. So if you want to have a programming exercise, you could figure out how this works. And you see from here basically what the API is, right? So format, all or names, what version of the database to use, what taxon it is in GenBank, right? And so you can write your own little script that will go through and generate this and pull it down for you, right? That's what you can do. So that's a very handy website not people really know about for getting stuff. A lot easier than just sort of, looking around on GenBank yourself. Okay. See how our script is going. Not well. Nope, so still not working right. So what's going on with that? Okay. Another approach people use for getting big. So if you've seen a big tree recently, like a tree of 20,000 species or something, how do they make it? No.
Stitch a bunch of smaller trees together.
No. So that's a good question. So it used to be people would do super trees. Right? So what's a super tree? Yeah, stitching them how?
Either attach nodes, just if you have a tree already resolved and you know what node goes with another tree, they would just go straight from the trees they have and just attach that way. Or you go back and just do a whole new matrix.
Right, so typically you do a matrix, right? So, and there's lots of actually approaches here. You can do triplets, you can do distance-based approaches. What typically people did was you have taxon A, B, C, D, and then another tree that's A, C, E, D, right? You encode this as a set of characters and say, okay, I have, A, state 1, B, C, D, question mark. A and B, 0, 0, question mark, right? This tree gives me A, question mark, C. It also gives me D and E, 0, 0, question mark, and so forth. So you get this, we call it a matrix representation of a phylogeny, MRP, right? So we convert these basically to a set of matrices for characters that would give you this tree under parsimony. And what you can do then is do an analysis using parsimony of this matrix. And you get some tree that sort of agrees with both of them or minimizes distance from both, minimizes changes from both of those. Okay, so you can build a super tree that way. It has some issues in various super tree approaches. People don't use it much anymore. Now they go for a different kind of approach, what we often call super matrix. Right? So rather than taking the individual trees and assembling them, they take the genes and use those. We're talking about like 18S and enolase. This might have 18S for all these taxa, and enolase for these taxa, and CO1 for these taxa, and heck, I could have morphology for these taxa. Right? Actually, morphology is more like that. Whatever. And so I could do this, and so this has all these gaps in it. Right? But as long as you deal with the heterogeneity well and have like different rates for each gene partition, you actually get a good tree this way. And so we had a tree published that was 94% gaps. Right? It's almost like a piece of lace, right? Where it's it has to have some sort of structure so it'll hold together. Your little doily on your countertop. There's many ways to have things that have that many holes that aren't a single object. But there are a few that will hold together, right? And so you have to make sure it's constructed in that way. Given how people make trees, it often is, right? Because everyone uses, okay, CO1's easy, let's get CO1, okay. I'm cool, I've studied this new, I'll work on this new gene. So you still have the basic genes for everything. I'll have my standard, taxon I use for everything, so I'll have some lines going across. So it tends to be that you have this structure, which you call a grove. Most people tend to use that sort of approach nowadays for building trees. There's also a third approach. Actually, Jeremy Beaulieu was involved in developing this, some sort of like llama-gram type thing, where it's sort of like manual super trees. We have a tree and you sort of hand like, let's put this together that way, and a ton of work, and no one uses that because a ton of work. Other approaches. So super matrices are used a lot. Yeah, go ahead. Yeah, this is super matrix. Yes. Yep. But it's pretty much how everyone does trees now. And the advantage of a super tree approach is that a parsimony search on a matrix like this is pretty fast, right? Whereas a search on a big matrix like this is 1,000 characters, this is 500 characters, and so forth, can be a lot slower, right? But that said, our tree inference programs are fast enough now. This is feasible, right? So there's a tree of I think it was 24,000 or so species that was run on it was a substantial desktop computer, but a computer, right? And so things can run fast. But now I'm starting out in phylogenetics. I mean, there's a famous data set called Zilla that was 500 taxa by I think one gene or something, and now it takes months to run. And like so, we came up with a new algorithm under parsimony. So with a new faster algorithm with the parsimony ratchet, and like in only 2 months, they got a better answer. Like, yay! But nowadays we run stuff. If it takes more than 2 days for parsimony, you're like, oh, why is it taking so long and stuff. And I just remember seeing in some of the videos. I mean, tree search is a very hard problem, right? So the fact that it's working so quickly is a bit ridiculous. So people now tend to use this. Yeah.
So when you run Flod,
Yeah, the getting to that. Yep, good. Yeah, so go ahead.
I was just asking if that was the same thing.
Yep. So Flod. So Flod is a way of getting super matrices and running it. So who's heard of Flod? Okay. So it's spelled because everyone likes PH. Right? Flawed, P-H-L-A-W-D, right? And it's a program that can go to so what you do is give it some base sequences. You can say, okay, here are my taxa I care about, green plants, mammals, whatever. Here are some sequences. So I'll take, a platypus CO1 and a echidna CO1, a kangaroo CO1, and a human CO1. Go find me sequences like that. And so actually, unlike Phylota, it actually uses gene names. It'll go to GenBank, make a little database of all these genes, and then assemble a supermatrix like this. Okay, and it has steps where it does the alignment for you, and things like that. And so a lot of the large trees you see, people aren't going up by hand and getting each species. They're running Flawed to suck stuff down from GenBank. Okay, why doesn't everyone do this?
Why? Trees?
Well, why? So we're having trouble getting treebase to work, right? ROTL we got to work this morning, but it didn't work yesterday, right? and then we're using someone's old stale trees. Right? Flawed, we can get a giant tree for, if I care about hibiscus, right? I can get every hibiscus in GenBank, and I can add my own sequences to it, right? So why not just do that all the time?
My experience with it, there wasn't a whole lot there from the taxa that I was interested in. So it was basically just one big opportunity.
Right. So if you work on uninteresting groups, there's not a lot of stuff in GenBank. So that's one aspect of it, right? So you still get your own stuff. Yeah. What else?
Well, if people do that a lot, that means that if you do that, you're creating a tree that already exists.
Right. So you can create this, 20,000 taxon tree that has 2 taxa more than the previous person's tree, right? You can just use their tree. Yep. And their expertise in if you want to date the tree, figuring out the time. We'll talk about this later in the semester. Figure out how to date a tree is really hard. You'll say this fossil goes here on this tree, constrains this. It requires a lot of expertise, right? And you probably don't have that expertise. Right? You might, but hard. Yep. What else? This is a practical thing. Flawed's super hard to install. Right? So in my lab, I know how to install things. I know how to debug things generally. And we have Flawed working on one computer, and that is the Flawed computer, right? And because it has these dependencies and things like that that are very tricky to install. So it's hard to use. Once you get it working, it's great. But until that stage, it's pretty hard to use. And once you do get it working, it still requires you to go through and look at your data. Okay. So one thing I want you to develop throughout this course is this instinct that when you get results, you have to dig into them and look at them, right? So with Flawed, oftentimes when you first run it, you'll find a tree, or something like this, and then right? And it could be that you've discovered that this member of this genus is super different from everything else on the planet, right? Or it aligned badly, right? Or it has only a tiny little sequence relative to everything else and just has lots of errors in it, right? Or it's a sequencing error. And so it requires you to go in and look at this to actually get the right answer. Now, if you just threw this from XML right into a dating program, you wouldn't see it. It would come back as an ultrametric tree. Yeah, it's a little longer than you expect, but whatever. It's fine. But you have to go in and look at your results. Okay. And so that sort of stage can take a lot of time. And once you also have to worry about biases, right? So if I get something that looks wrong to me, it could be just I was wrong about the biology, right? Maybe that isn't a clade. Right? Maybe dicots aren't a thing. Maybe they're not a clade actually, right? We know they're not. And so you have to then revise your thoughts that way. But could also there's the risk that you would then manipulate it until you get something that looks right to you, even though it's wrong, right? Anytime you manipulate data, you have to worry about that. But also times lots of data are wrong, right? They're mislabeled in GenBank and things like that. So you have to be careful. And so doing this well requires a bit of art and looking at it for a long time. Yeah. Questions about that?
Did you say dicots aren't a clade?
Yeah.
Huh. I don't know. It's because dicots are so,
Eudicots are clade.
sure.
The true dicots.
Right. Yeah, exactly.
Yeah.
That's why I was confused.
Oh, really? Huh. Yeah. So we discover things. And, but none of them are called basal. Yeah. Okay, so we're still having issues here. So cannot open connection. So what we can do now is look at this.
Oh, so what are we doing? It failed again?
It failed again.
Within that function, though?
Yep. So let's see what happened here.
Can Can you? Is there a way? So that check you did earlier. Is there a way to check the last time that it worked? Like what state it was in?
No.
Will it change since that state?
No. I don't think that's stored in the Travis thing. And actually, what's more likely in this case is that treebase changed rather than the code change. If you look at the code.
Since it's last bit, you can see its last version.
Yeah. Last touch 2 years ago.
Yeah.
Right? And so something probably changed in treebase since then. Yeah. And that's one thing. I mean, so even like the Open Tree of Life API, they have a version 1 and version 1 and version 2, right? And talk about things like breaking or not being backwards compatible. That's what that is, right? So, for example, the programming language Python. Python 2.4 or 6 or something is used by certain people, and Python 3 is used by other people. And the Python 3 code won't work in Python 2.6 and vice versa, right? So they broke that backwards compatibility. When Perl 6 comes out, we'll have the same issue. And so that happens. There's a big case in phylogenetics actually with ape. Ape changes phylogeny structure internally. And so all the other packages that use ape's phylogenies now start getting errors and stuff like that. So actually in ape, even today, there's a function called new to old philo and old to new philo. You can convert between those two formats. All right, so I'll spend a few more minutes debugging this and then sort of give up. All right.
So the homework was supposed to work.
It was supposed to work, and when I checked, it had worked. Right. And this would be way overkill for you, right? But then again, if you're trying to do something where you need the data, yeah, it's important.
Yeah, I had the running theory that it was supposed to miss that.
No. I might put that sort of thing in, but not this bad. And I'd probably give you a clue like, dig into the source code for this and figure out what, not just like, yeah. Okay.
If you don't like someone, you can give us a task. Go contact the developer and ask him to fix this and that.
Yeah, or her. Yeah. And one issue about code is that, like, so once I publish a program, right, people start using it. Do I get a lot of benefit from helping people? Probably not. I might get some more citations, but not a lot. Not as much as I would get from like pushing a new thing, right? Not dealing with the little corner cases. And so a lot of phylogenetic software, it's hard to get it updated. I mean, so example is Versatree, right? It's a cool very cool package, very useful, right? It's not being updated much anymore. And actually, for a while, it's at risk of being thrown off of CRAN. Or Laser, the same thing. Laser actually was thrown off of CRAN for a while. It's a program for looking at diversification. And then people said, oh, we need Laser. And so it was put back in. But, it's very easy for software not to be supported anymore.
So, in a case like that, if Laser's on GitHub, if I wanted to fix it because it was kicked off CRAN, then I could fix it, send a pull request to them. They could look it over and say, all right, fine.
Yeah, exactly. Or you could say, I just want to take it over and run it from my GitHub account. That can work too. So, for example, RCPP, which is this R interface to C++, a programming language. I was in a hackathon and we were using it, and it had been basically killed for most of CRAN, except for this one Italian repository that was out of date. Hadn't yet killed it. And so we took it from that repository and stuck it in our code and used it. And later, some people started taking it over and actually started using it a lot. Now it's used in, it's huge, like a quarter of all our packages use it, or some huge number. And so now it's really being developed well, because someone took it over from the guy who worked on it and then left. So that's possible too. Okay, let's try this. Woohoo! So this part worked. And again, this looks hard to read at first, right? So what am I doing? I'm sort of skimming through to see information it has in it. Especially like, yeah. So like, it has a link to something there that looks interesting. So various studies and so a study in a tree, I believe, So we can dig through that way. And now I can go back to the code and see, okay, so page 1 is that. Go back to my test code. Okay, so I have that. And go up here, and the next step after it gets that is use XML hits. So it parses the XML to a structure, which means it goes through, so XML is like a nested thing, and it walks through that. So let's look at that. That worked. Okay, now it's a little easier to read, right? Okay. Rockin'. Here's where it should fail. So max trees equals 1, let's say. Let's run this, see what happens. Resources. Yep, that works. Has a tree in it. Good. Metadata. Yep, that's tough. What does this line do? Yep, exactly. Gave us 2 studies. Good. Gave us 2 trees. So we're rocking this, getting everything right. Got that. This is a sort of warning message, but that's actually okay. All right, so it's still working okay.
Why was that message okay?
It's just how it opens and closes connections to the web. It's a message I've seen before without it causing an issue. Okay, now here's the thing it's trying to do. Here's probably where the error is. So I see that it requires arguments. All right. So looking at those arguments. Yep, so again, digging deep inside. Almost there. So it's getting lots of the information. Okay, so here's the issue. So let's look at try recursive. That's a very simple function, right?
But scan's still hidden somewhere else.
Yep. So it's probably in treebase dig. Yep. Probably right here where the issue is. Yeah. So digging that's going to take a little, let's see.
So as the person that made treebase probably getting email knows about it, so he's sitting somewhere doing the same thing too, he or she.
I don't think it's a T. If people use it a lot, yes. It probably isn't used very much, actually. So how most people interact with treebase is going here and going to the web and either looking for papers that look good, right, and then download the relevant tree, or just knowing that like, oh, that Pyron paper has a great tree. Where's that stored? Oh, it's on treebase. Let's go there and get it. Yeah. Yeah. Yes, that's what it's trying to do. So URL is trees1. That worked too. Yes, that's the issue. So here, yeah, so I could dig into more, but it looks like here's the issue, is this URL thing. They keep digging down into the code. And so basically, if you look at the structure, and there's a different way to go. I'll show you a different way to go after this too. But first, you look at search treebase, I think was the first function. And inside there, it was eventually to get URL content, and then so forth. So keep digging down to finding it. Now, another way to handle this sort of thing, perhaps easier, is debugging within our options, and you can do options error equals utils dot recover. Okay, and what that does is when I have an error, it will drop me right there where that error occurs. Okay, so we can go all the way back to this function. Try this. Let's see if it fails properly. Nope.
So you don't apply that options, the error thing to a specific try, if you just do it and any function you run after that, it will do it?
Yep. Except I think these functions are wrapped in try, right? So try makes you invulnerable to errors. Otherwise it would have stopped. But had I done, so now if I have like some function like, how many trees 2, right? And I do function n, and I say return how many trees, which is not a real function. That's fine. I'm not going to run it yet. Now if I try running it, it shows where the error is. Right. Try it again, but now I'll pass it something that makes no sense. it now shows me these different levels. So I have my function. My function calls ape's function. Ape's function calls this. That calls this. And that's trying to go from up to n, right? And where n is cat, right? So that's where the error is. So it digs me right into where the problem is first.
So this error that we got with Aaron's scan, when you ran the
Yes.
so that's the error that we were getting though. So
Right.
why even run this instead?
Oh, so the problem with this is since that. So if you look at the code here. See right here? See that try? Try is a function that normally if you're running R code and has an error, it'll just stop, right? Which is often what you want to do, right? Trying to do a phylogeny and like, oh, it didn't load the data well. I'll keep going anyway. Like, no, stop if it has an issue. And what options recover thing allows you to do is if you have an error, it goes in there and look at it, right? But something like Gazer from the website, you can imagine, the website's down for that taxon, or there's no data for that taxon. The next thing's gonna be fine. By putting it in try, it will just like try, get an error, and keep going on. And so rather than stopping here, it keeps rolling on. So the options recover doesn't stop it. Yeah. Okay. So I can dig into this more and try to figure out what's going on. But you have the basic idea, right? Now what I'd like you to do is talk about. So you talk about like you each have to come in with some projects you're thinking of, and start thinking about talk about like what you want to do for your project. Yeah, we didn't have to talk about that yet, right? So go ahead.
So specific projects that we want to put in this course.
Yep. Stop recording this.